Okay, everybody, so this is kind of what I would probably say to you in class about Chapter 16, Adaptive Immunity. I've changed these slides a little bit compared to the ones that were originally posted in the PowerPoint lecture tab in eCampus. So I will upload this version as well. I'm just letting you know that these are a little bit different. I've deleted some slides that aren't really relevant to us, and I've also added some slides from my A&P 2 class because I think they actually get some of the points across a lot more clearly compared to our book. So you might want to use these notes on your own as you're listening to this lecture as well. I'm also going to use the immune system outline that is posted in the study guide tab of eCampus. So you're not going to be able to see that, but I'm referring to the outline as I'm going through these slides. So as you're preparing for this chapter and reviewing for this chapter, I would do the same thing. Have the book open, have the slides open, and have that outline open as well, and take notes as you need to. All right, so the immune system in general, this is basically everything that we have in our bodies that are designed to recognize foreign cells like bacterial cells or virus infected cells or things like that, recognize something foreign and get rid of it basically. And so chapter 15 focused on the innate immune response, and those are things that have already been set up in our body and are ready to respond no matter what the foreign invader is, whether it's a virus, whether it's a bacteria, and no matter what type of bacteria, those innate immune responses are going to respond always and in the same way, no matter what the invader is. It could even be just tissue damage, like a cut or something like that. But the adaptive immune system is a little bit different. Most of the things that are part of this immune system are in our blood, so it's a lot more internal. We're not talking about skin as a barrier in this sense. That's for innate immunity. And the components of this immune system are designed to respond to specific invaders, and the responses might happen a little bit differently depending on what the invaders are. So some of the key characteristics of the adaptive immune response. So specificity, I kind of already talked about that. We're talking about a specific invader. Inducibility, I'll talk about that in a second. Clonality, unresponsiveness to self, that's very important. We don't want our immune system to respond to our own cells. We want it to respond to foreign things. And then memory. So in terms of inducibility and clonality, these have a little bit to do with specificity as well. So very generally, let's say, I'm going to just use an example of an invader. Let's say chickenpox virus. So there's something very specific, maybe a protein or something like that virus produces. And when it infects our cells, that's what your immune system sees. And that's what we call an antigen, which we'll get to that in a second. Your body and our bodies, all of our bodies have components in our adaptive immune system designed to respond specifically to chickenpox virus. But we might only have, let's say, 10 immune cells at birth that are designed to respond to that virus. And so if you never are exposed to that virus, you will never use those specific immune cells. But if you are exposed to that virus, now we have a specific adaptive immune response using those specific 10 adaptive immunity cells that are designed to respond to the chickenpox virus. They will be induced to respond. That's where inducibility comes into play. And then part of the immune response means your body is going to make more of those chickenpox specific immune cells. And that's what clonality means, meaning we're cloning cells, making more copies of the cells that are designed to respond to that specific invader. Not only are you going to make immune cells for that specific and current infection, but you're going to make, during this process, you're going to make memory cells that are designed to respond to the chickenpox virus in case you are exposed again. So the benefit of this adaptive immune response is not only can you fight the current infection, but you're left over with a lot more cells now, more than 10, maybe 1 million now. And these numbers are not accurate. I'm just using them as illustration. But maybe now, instead of just 10 chickenpox immune responsive cells in our bodies, maybe now we have 1 million. So that if you are exposed again to that same virus, the adaptive immune response can respond more robustly and more quickly so that you don't get sick. And that is kind of the basis behind vaccines as well. So what I'm going to talk about are some of the, I'll give you a little bit of an overview and then talk about some of the key players. That's kind of how I organize the outline. And then we'll get into the 2 specific types of adaptive immunity. So the cells, the main cells that are coming into play during the adaptive immune response are lymphocytes. So we have B lymphocytes or B cells, and we have T lymphocytes or T cells. Both of these types of lymphocytes, these are a type of a white blood cell, so they are both produced in the bone marrow. But in order for them to be able to respond in the immune process, they need to mature. And where they mature is why and how they're named. So B cells will stay in the bone marrow and mature there. T cells will leave the bone marrow and go to the thymus, and that's where they mature. These play roles in the 2 types of adaptive immunity. So the 2 types are cell-mediated and antibody-mediated, or the humoral response. So we're going to get into those details in a second, but keep in mind that a lot of the key players that I'm going to talk about in a minute are, they're going to play a role in both of these responses. Some are more specific for one response versus the other, but the main steps that have to occur is going to be the same for each of these responses. So that'll be more toward the end. So let's talk about some of these kind of key players. So T cells or T lymphocytes, produced in the red bone marrow, they mature in the thymus. They're going to circulate your lymph and in your blood, and they'll set up camp in your lymph nodes. And like I had mentioned earlier about my chickenpox example, all of our T cells have specific receptors on their membranes that are designed to recognize and bind to very specific invaders, specific bacteria, specific viruses, fungi, whatever. Let's see. So this is kind of what we would look like, what that T cell receptor would look like. On the surface of the T cell, this is where that receptor would bind to the foreign invader. The types of T cells. This book lists 3. The 2 that we're going to be most concerned with are cytotoxic T cells and helper T cells. So cytotoxic T cells do exactly as they're named. They are toxic to cells. They're going to directly kill other cells. So maybe that means infected cells or, let's see, foreign transplant cells, things like that. And then there are helper T cells, and these help both of those 2 types of immune responses. So you need your helper T cells for both that cell-mediated response and for the antibody or humoral response. And we'll see what role exactly helper T cells play in a minute. B cells, so these are found in Well, they're made in the bone marrow. They mature in the bone marrow. They're going to leave once they're mature and set up camp in some of our lymphatic tissues, like your lymph nodes. And basically, those B cells and T cells, I think the easiest explanation is just to imagine them hanging out in your lymph nodes and waiting for invaders to show up. And it has to happen a little more elegantly than that. And we'll get to that in a second. So they're just waiting there. And when they recognize that there is an invader in the blood, then they're going to respond. B cells you should equate with or associate with antibodies. So when we make antibodies, that really is the job of the B cells. And we'll see where and when that has to happen when we talk about those two types of immune responses a little bit later. This is what a B cell receptor would look like. So just like in the T cells, the B cells have receptors on their surface that are designed to bind to a specific foreign invader. I want you to remember this Y shape of a B cell receptor when we come back to antibodies. All right, so what exactly is your immune system responding to? So I keep saying foreign invaders, foreign cells, bacteria, viruses, what have you. The reality is the specific thing that your immune system is responding to are antigens. So when you say that word antigen, we're talking about a molecule like a protein or a piece of a molecule, a piece of a protein that your body recognizes as foreign and worthy of attack. And so all of these foreign invaders that I'm talking about, viruses, bacteria, other types of microbes, think of bacteria specifically. Those are cells. They have a cell membrane, they have a cell wall, and they have proteins on their surface. It's one or a few of those proteins on the surface of the bacteria that is going to be the actual antigen that your body recognizes as foreign. Okay? So this is showing you, so this term epitope is kind of like the more specific part of the antigen that your body is recognizing. Sometimes I use the term antigen and epitope interchangeably. It's probably not technically correct, but for our purposes, we can kind of use those terms interchangeably. Epitope is on an antigen, and it's the antigen that's on the surface of the pathogen. That's what your body is responding to. There are different types of antigens. So again, I'm using the outline, and I try to rearrange these slides so they kind of match up with the order of the outline. So exogenous antigens, these are things, they could be toxins, like bacteria can produce toxins. It's the toxins that could serve as the antigen. It's exogenous, meaning it's not part of the cell. Could be on the surface of a cell, something like that. So bacteria would probably more often than not have exogenous antigens. Endogenous antigens are those that are produced by the microbe when the microbe is already inside your cell. So if your cells have already been infected, then the antigens from that microbe that has infected your cell, those are endogenous because they're sort of coming from inside your body cells. Autoantigens, those are, I kind of consider those as self-antigens. These, so when I was originally talking about this term antigen, you're talking about something on the surface of a foreign invader, a foreign microbe. But our own cells have these little flags on their surfaces too. And part of the immune system's job, which we're not going to go into a lot of detail about, but understand that this has to be the case, not only does your immune system have to respond to foreign antigens, but it also has to recognize your own antigens and know that, oh, okay, these cells belong here. I don't want to attack those. And so it's important that we do have these autoantigens. So your immune system can tell the difference between what is a foreign cell or an infected cell and what is part of you. Here's just some examples of those 3 different types of antigens. One particular type of autoantigen or self-antigen is this major histocompatibility complex. So this specific self-antigen, I'm going to skip ahead a couple slides because I want to use these slides that I added to this PowerPoint session. So major histocompatibility complex proteins, those are self-antigens. Their job, one, is to tell your body that they belong to you. Tell your body that the cells that have these antigens belong to you. But also, their job is to hold on to and serve as like a handle for foreign antigens. So let's look at this in a little more detail. So this is showing you, again, it just looks like some kind of receptor on the surface of a cell, but this particular type of protein, 1, telling your body that this cell belongs to you, but 2, also serving as a handle to hold on to foreign antigen. Why do we need to do that? And I'll talk about that in a second. So these 2 slides right here, I really like these. These are from a different book. They're from my A&P class, but I think it really gives you a much clearer description of the job of these MHC proteins and the difference between the 2 classes. So there are class 1 MHC proteins and class 2 MHC proteins. So class 1, let's look at those first. Where do you find these? They're on the surface of all of your cells, all nucleated cells. So that means no red blood cells. So they're on every cell except your red blood cells. Its job is to bind to and hold on to endogenous antigen. So it's going to serve as a handle for that endogenous antigen that's coming from a foreign invader. We need class 1 MHC proteins in order to kind of activate, in a way, cytotoxic T cells. If you see CD8 here, equate that with cytotoxic T cells. CD8 is another term for that. They mean kind of the same thing. So when you're, I think I should have skipped over to something else in a minute. I'll get to that in a second. So when that MHC1 protein, which is on one of your cells, and it's holding on to a foreign antigen, that is when a cytotoxic T cell will be able to respond to it. I'm going to come back to this in 1 second. Let me talk about class 2 and let me talk about antigen presentation, and then I'm going to come back to this slide. Class 2 MHC proteins, again, the job is still to hold on to foreign antigen, but where do you find these? You find these on, not on every cell, but on antigen presenting cells. And again, they're serving as a handle. You need them in order to activate helper T cells or CD4 cells. Okay, so let me talk about antigen presentation first, because it makes more sense why you need the MHC proteins. So in order for those T cells and B cells that I said were hanging out in the lymphocytes, or in the lymph nodes, in the lymph nodes, they're hanging out in the lymph nodes. In order for them to even recognize that there are foreign bacteria floating around in your blood, or virus or something like that, they need to be formally presented with those foreign antigens, almost like as if they're like a king in court and they need to be presented. Their guests need to be formally presented to the king and queen. It's kind of like that. So you can't just have random bacteria and random virus floating around and getting into your lymph nodes and expect your B cells and T cells to recognize and respond to them. They will only recognize foreign antigens if those antigens are formally presented to them, T cells and B cells, through an antigen presenting cell. So antigen presenting cells will be kind of going out through the blood, surveying the blood, looking for foreigners, and their job is to go find the foreigners, engulf the foreigners, and display the foreign antigens on their surface, and then find their way to a lymph node and find the right B cell or the right T cell and say, hey, look, this is in the body. We need to do something about it. So that's the job of an antigen presenting cell. What can be an antigen presenting cell? 3 types. Dendritic cells. This is a pretty effective antigen presenting cell. Macrophages, and then sometimes B cells themselves can serve as their own antigen presenting cell in a way. Okay, so now that we know that T cells and B cells need to be presented with foreign antigen by these antigen presenting cells, then it makes more sense why we need class 1 and class 2 MHC proteins. So MHC1 proteins are going to be responsible for activating cytotoxic T cells, whereas class 2 MHC proteins are responsible for activating helper T cells. And you find these proteins on the surface of cells. So class 1 MHC proteins are on all cells. The message that the class 1 MHC proteins are telling depends on what the cell is that is holding onto the antigen through its MHC1 protein. So let's go down here. So all of our cells except our red blood cells have class 1 MHC proteins on their surface. They are all capable then of holding on to endogenous, let's go back here, class 1, endogenous antigen from a foreigner. If the cell that has the class 1 MHC protein that is holding on to foreign antigen, if this cell is an antigen presenting cell, then the message it's sending is this: I belong to self, meaning I'm an antigen presenting cell in this body. I've captured a foreign invader. This is what it looks like. Kill any cell that displays it. So this is going to be part of the process of activating a cytotoxic T cell, because the cytotoxic T cell's job is now going to be go and find more of these antigens and get rid of the cells that are making them. If this cell up here is not an antigen presenting cell, then that means it's an infected cell. So cells that are infected will display, let's say it was infected by a bacteria. Well, let's say virus. Let's say a cell in your body is infected by a virus. That cell, that infected cell, is going to display that viral antigen on its surface, and it's going to do so using its MHC1 protein. So now we have an infected cell that is displaying viral foreign antigen on its surface using a class 1 MHC protein to hold on to that antigen. Now the message is a little bit different. The message is, I belong to this body, but I've been infected. or maybe I become cancerous. You need to kill me. So in this case, the message is once again to a cytotoxic T cell, but to one that's already been activated. So the point here is that in order for an immune system cell to recognize the foreign antigen, it has to be displayed on the surface of one of your body cells through one of these classes of MHC proteins. Let's move to MHC2 proteins again. So these proteins are on the surface of all of your antigen-presenting cells and only antigen-presenting cells. And so in this case, there's only one message that this type of cell can say, meaning I belong to this body, but I've captured a foreign invader. This is what it looks like. Help me mount a defense against it. This is what's important. Why does it say help me mount a defense against it instead of over here where it says kill any cell that displays it? That is because in this situation, the antigen is being held by a class 2 MHC protein. Class 2 MHC proteins can only talk to helper T cells, whereas class 1 MHC proteins can only talk to cytotoxic T cells. In this case here, if we're talking directly to a cytotoxic T cell, we're telling that cell you need to kill something. But if we're talking to a helper T cell, we're saying we need help. We need help gathering in a response against this particular antigen. Okay? These messages are going to come into play when we talk about those two types of adaptive immune responses. I know I've talked a lot already and I've given you a lot of detail, but just remember the overall picture is if you are infected with something and your adaptive immune response is responding to it, it's either responding using the cell-mediated response or it's responding using the antibody or humoral response. One or both of these messages here have to happen in order for that response to occur. Okay, so keep this stuff in mind when we get to those two specific types of adaptive immune responses. All right, so what have we talked about? We've talked about the two different types of lymphocytes. We've talked about the fact that there are two types of T lymphocytes. We talked a little bit about antigens, and then we talked a lot about these MHC proteins and why they're important. They're holding on to those foreign antigens and allowing the right cells in the immune system to recognize the antigen and respond properly. All right, so we talked about antigen presentation. Oh, antibodies. Here is another player in the immune response, specifically in the antibody-mediated response. So antibodies are also known as immunoglobulins. They are proteins. They are made by activated B cells. Specifically, they're called plasma cells. So one thing I didn't mention earlier, but if you recall when I was giving you the very first overview of the adaptive immune response, I had said that during the adaptive immune response, either form, what's happening is you have some specific cells, T cells or B cells, designed to respond to that specific infection, and they will fight the current infection and then create more of themselves that will hang around as memory cells for the next time you're exposed to that particular infection. The cells, the lymphocytes that are currently fighting the infection are called effector cells versus the memory cells that are made for the next time you're exposed. Plasma cells are effector B cells. So when a B cell gets activated and starts doing its antibody-mediated response, it's going to turn, some of those B cells turn into plasma cells, which are responsible for making the antibodies. And it's the antibodies that are going to deal with the current infection. An antibody, remember I said earlier, take a note of what that B cell receptor looks like. This is basically what an antigen is. What an antibody is. It is the B cell receptor. So if you have, let's say, you have 10 B lymphocytes in your body that have a receptor designed to respond to strep, then when those 10 cells are activated because strep has gotten into your system, the end result of the antibody-mediated response is to make a whole bunch of those B cell receptors that are on their own, and they can go into the blood and go find more strep. The B cell receptors are the antibodies. And if the B cells that are being activated are the ones that are specific for strep, then the antibodies they generate will also be specific for strep. What do antibodies actually do to help fight the infection? So these are the functions of different antibodies. So they're that Y-shaped protein, and their job is to bind to a specific antigen. Well, what do they do when they bind to the antigen? So they listed some of the functions of antibodies here, but I'm going to use the picture to describe some of them. So neutralization. Let's say the antigen is a bacterial toxin. Well, the antibodies that are going to be made in response are designed to bind to the toxin. So if they can bind to the toxin, they can help neutralize that toxin. Maybe they can neutralize the bacteria if the antigen is on the surface of the bacteria. Sometimes they can cause agglutination, which is clumping. If you've ever done blood typing in A&P2, this is what was happening. Antibody and antigen that were designed to bind to one another did so and caused clumping. So in this case, they're showing you antibodies designed to bind to specific bacteria through the antigen on the surface of the bacteria, but when they do so, they cause other bacteria antibody components to clump together. And that's another way of when you cause clumping of the bacteria, you're kind of preventing the bacteria from invading other tissues, and also in some ways making it a little bit easier for phagocytes to find the bacteria, because now you have this big clump of bacteria. Opsonization is a fancy word for flagging. So one way, and this has to do with phagocytosis. This particular bacteria has lots of antigens on its surface. When the antibodies were made in response and they go out in the blood and they find those antigens, they bind and they serve as flags on the bacteria. The flag is a signal for a phagocyte to come in and engulf this bacteria. And then antibody-dependent cell-mediated cytotoxicity. That's a mouthful. So in this case, antibodies were almost in a way serving like a flag and a way to invite natural killer cells, which are part of the innate immune response, to come in and kill the actual bacteria. So the one thing I want to point out here is these are some of the functions of antibodies. Notice that the antibodies did not directly kill anything. They just made it easier for other processes in our body and other cells to kill and get rid of the invader. That's different from cytotoxic T cells that are part of this T cell population, and we'll see that in a minute. Those are produced during an immune response, and they will actually kill other cells. Antibodies don't kill other cells. They just make it easier for other things to kill those foreign cells. Okay. This book also talks about the different classes of antibodies. I'm not going to go into detail about that, but just understand that there are different classes, and this is where all these IgM, IgG, IgA come into play. Remember, antibodies are immunoglobulins, and that's what the Ig stands for. Different classes of antibodies can be produced by the same immune response. So the same foreign antigen could trigger the production of different classes of antibodies that are all designed to bind to that same foreign antigen. Some are produced more quickly and more robustly, and might be produced due to a specific type of infection versus others. I'm not going to go into detail. I'm not going to ask you details about these different classes. Just understand that there are different classes of antibodies. All right, the last key player. I haven't even gotten to the immune responses yet. Can you believe how complex this is? I cannot. So the last player in the adaptive immune response are these chemical signals called cytokines. So different leukocytes or white blood cells will produce cytokines as part of these adaptive immune responses, and they are a way to communicate with other cells. We'll see that in a second. Here are some of the different types of cytokines. Interleukins, so they're signaling among different leukocytes or white blood cells. Interferons, they're going to play a role in viral infections. Growth factors, tumor necrosis factor, and chemokines. Just understand and know that these are the 5 types of cytokines listed in our book, and just understand that they are responsible for cell signaling and communication among different cells during these immune responses. All right, so that was a lot of information, and I really was just talking about some of the common elements in these 2 different adaptive immune responses. Okay, so we know we have to have lymphocytes. Which ones? We'll get to that in a second. We know we have to have foreign antigen to begin with. We know we have to have class 1 and or class 2 MHC proteins in order to help present those foreign antigens to the lymphocytes. We have to have antigen presentation. We have to have antibodies sometimes, not in all responses, but in some responses we will have antibodies, and we need to have cytokines. So all of these things are playing a role. So now let's talk about the 2 different adaptive immune responses. So I said earlier, and if you look at the very beginning of the outline, it outlines it for you. What are the 2 types of adaptive immune responses? The cell-mediated response, and then in a few minutes, we'll talk about the antibody-mediated response, also known as the humoral response. Okay? All right, so let's look at the cell-mediated response first. This is a response against intracellular pathogens and abnormal body cells. So specific, intracellular pathogens are more often than not viruses, because viruses can't exist on their own, right? So when you're first invaded by a virus, yeah, there might be a short amount of time where there's going to be free-floating virus in your blood. But it can't exist like that for a long time, and the virus wants to infect one of your cells through those processes that we talked about when we talked about viral replication. So more often than not, if you have a viral infection, the virus has already invaded and infected your cells. So that's what we mean by intracellular pathogen. The pathogen is already inside your cells. Other types of kind of like intracellular invaders would be abnormal body cells. So cancer cells are abnormal body cells. Foreign tissue transplants, those are body cells, but they're not your body cells. They're abnormal. They're from somebody else, but they're still body cells. And so human cells. And so these are the types of things that the cell-mediated response is going to respond to. I always, and for our purposes in this class, I think it's safe to do this. If you're talking about a viral infection, it's the cell-mediated response that will respond. If you're talking about a bacterial infection, no, because more often than not, bacteria are free-floating in your cells. They have not really infected a body cell. They might, and if they do, then the cell-mediated response will respond. But more often than not, that is not the case. So I think it's safe for us right now in this class to say viruses are going to cause a cell-mediated immune response. Bacteria will cause an antibody-mediated response. And we'll look at that in a minute. Okay, so what's happening in the cell-mediated response? So the goal is we need to activate cytotoxic T cells so they can go leave the lymph node, go out into the blood, and find all of those infected body cells and get rid of them. And there's 4 things that have to happen in order to activate a cytotoxic T cell. These 4 things are also going to have to happen in order to activate a helper T cell and when you need to activate a B cell in the antibody-mediated response. So even though these 2 different responses will be triggered by different types of antigens or invaders, the same basic things have to occur. And what are these things? Antigen presentation. So a cytotoxic T cell, it is hanging out in the lymph node. It is waiting for an antigen presenting cell to get to the lymph node and say, hey, I'm holding on to a foreign antigen, an endogenous antigen, through my MHC1 protein. This is what it looks like. You need to go and kill it. We need helper T cell differentiation. We cannot activate cytotoxic T cells without helper T cells, and I'll show you why in a second. Clonal expansion. This means we need to activate the cytotoxic T cells and tell them to make more copies of themselves, because remember, we don't have very many. Let's use our chickenpox virus as our example again. Let's say we have the chickenpox virus. We only have, and again, this is just for an example, this is not a true number, but to illustrate my point, we only have 10 cytotoxic T cells designed to respond to chickenpox virus. You can't fight an infection with only 10 cells, so you need, part of this activation process means making more cytotoxic T cells that can respond to chickenpox. That's what clonal expansion is. Self-stimulation basically is talking about the ability of activated cytotoxic T cells to cause activation of even more. All right, so this is what this looks like. So we have antigen presentation. This is what I want you to see. Here is a dendritic cell. A dendritic cell is an antigen presenting cell. So because it's a cell with a nucleus, it has MHC1 proteins on its surface, but because it's also an antigen presenting cell, it has MHC2 proteins on its surface. Both of these are capable of holding onto, I'll use our chickenpox example again, they can both hold onto the same chickenpox antigen. And the messages they are sending, they're being sent to 2 different types of cells, and the messages are different. So if we look at MHC2 protein, MHC2 proteins can only talk to helper T cells. Here's our helper T cell here. It is inactive. We need to activate it. So what does activation of a helper T cell mean? It means this helper T cell has to bind with its receptor. It is designed for chickenpox antigen. So it can bind to that chickenpox antigen that's being held by the dendritic cell through its MHC2 protein. And the message that this binding event says is the dendritic cell is saying, I've encountered an invader, chickenpox virus. This is what it looks like. Help me respond to it. And that's how we activate the helper T cell. So what happens is that helper T cell, first it binds to the antigen, and then it needs to be co-stimulated in order for it to fully be activated. Co-stimulated by a cytokine. In this case, interleukin 12. Do you have to know interleukin 12? No. Just understand that in order to activate the helper T cell, first it has to bind to the foreign antigen through the antigen-presenting cell, and then it needs to be co-stimulated by a cytokine. In this case, the cytokine is coming from the dendritic cell itself. So we fully activated the helper T cell, and what happens? It differentiates or turns into a slightly different helper T cell, a type 1 helper T cell. The job of this type 1 helper T cell is to release another kind of cytokine, and the job of this cytokine is going to help in this process over here. So what is this process over here? Nikki, this is how we're trying to activate our cytotoxic T cells. So the same antigen-presenting cell can present the same foreign antigen to a helper T cell and a cytotoxic T cell at the same time. In this case, this dendritic cell can talk to an inactive cytotoxic T cell designed to respond to chickenpox through its MHC1 protein. And here we're looking more closely at that binding event. Here's the MHC1 protein, here's the chickenpox antigen, here is the T cell receptor on the inactive cytotoxic T cell. We need to activate it. Activation means binding to the foreign antigen and co-stimulation. Here is why we need the helper T cell. We cannot co-stimulate this cytotoxic T cell without interleukin 2, without this particular cytokine. This cytokine is coming from the activated helper T cell. So this is why we call it a helper T cell. It is helping to activate the appropriate cytotoxic T cell. So here's our activated cytotoxic T cell. Now it's going to undergo what we call clonal expansion. That means it's going to make more of itself, more active or effector cytotoxic T cells, and then some memory cytotoxic T cells for later, in case you're exposed to chickenpox again. The active or effector cytotoxic T cells are self-stimulating. They're activated, they can release their own interleukins or their own cytokines and cause further expansion or cell divisions to create more and more copies of cytotoxic T cells that are active and can recognize the original antigen. In this case, we're using chickenpox as an example. So what are these active cytotoxic T cells going to do? They're going to leave the lymph node. All of this was happening inside a lymph node. They're going to leave the lymph node, go into the blood, and start looking for more cells that have been infected with chickenpox and are displaying the chickenpox antigen on their surface. That's how the cytotoxic T cell knows that that's an infected cell, and it will get rid of that cell. How does it do that? Two different pathways. We have the perforin granzyme pathway and the CD95 pathway. The end result of both of these pathways is death or apoptosis of the infected cell. It just happens a little bit differently. So in this perforin granzyme pathway, when the activated T cell It goes out into the blood. It finds an infected body cell, which is displaying that foreign antigen on its surface, and they bind to one another. That binding triggers the release of perforin from the cytotoxic T cell. Think perforin, think perforate. It's poking holes in the infected cell. And then granzymes come from the cytotoxic T cell, which are special enzymes. They can get into the infected cell through the perforin pore and trigger apoptosis or cell death. The CD95 pathway, and again, when is which of these pathways used? To be honest, I'm not 100% sure, and we don't need to know that information. That's more detail than what you guys need to know. Just understand there are two different ways that a cytotoxic T cell can cause cell death. In this situation, again, activated cytotoxic T cell has left the lymph node, gone into the blood, and found an infected cell with the same foreign antigen that activated itself. It binds, and then in this situation, there's another binding event that leads to apoptosis of the infected cell. So what do we have? In the cell-mediated response, let's go back to look at this picture. An antigen-presenting cell presented foreign antigen from either a virus or an infected, I would say an abnormal body cell, like a cancer cell or something like that. Presented the foreign antigen to both helper T cells and cytotoxic T cells with the right receptor. And in doing so, activated both helper T cells and cytotoxic T cells. Activation of helper T cells has to happen first because we need the T cell to help activate the cytotoxic T cell. Helper T cells help activate cytotoxic T cells. When the cytotoxic T cell was activated, it created more copies of itself. Active effector cells that will leave the lymph node, go into the blood, and kill infected cells, and then also created some memory cytotoxic T cells. So at the end of this, at the end of this initial immune response, I used those numbers to illustrate this point earlier with the chickenpox example. If you had 10 cytotoxic T cells to start with, designed to recognize chickenpox antigen, at the end of this initial cell-mediated response, you might end up with now 10,000 memory cytotoxic T cells, all designed to respond to chickenpox virus. So if you're exposed again, this happens much more quickly, faster than it would take for you to even feel sick. Okay, that's cell-mediated response. The antibody-mediated response happens very similarly. The same basic things have to occur. The biggest difference is what types of invaders does the antibody-mediated response respond to, and what is leaving the lymph node to deal with the invaders. Okay? So in the antibody-mediated response, this is responding to exogenous pathogens and toxins, meaning not infected body cells, but just free-floating cells. So those are going to end up being, and not body cells, something different. So bacterial cells, and maybe bacterial toxins. These are exogenous. Now, I had mentioned earlier, we were talking about the cell-mediated response, and I was equating that with viral infections. And I said, when you're initially infected by a virus, there might be a short amount of time when the virus is kind of free-floating and trying to find the cell it wants to invade. During that time, the antibody-mediated response may be able to respond to that free-floating virus. But once the virus has entered one of your own cells, the cell-mediated response will take care of it. And more often than not, if you have a virus, it's going to be in your cells already. And so that's why it's usually the cell-mediated response that will respond. Okay, so now we're focused on the antibody immune response, also known as the humoral response, and we're talking about bacteria. All right, same things have to happen. Antigen presentation to a B cell in this case. We also need to activate helper T cells. And then those B cells, once they're activated, they're going to do the same thing that the cytotoxic T cells would do. They're going to undergo clonality or proliferation and differentiation. They're going to make more copies of themselves. They're going to make effector B cells or plasma cells that are going to deal with the current infection. And they're going to make memory cells for later. What's not shown here is, well, where do antibodies fall into this? And we'll see that in a second. Let's look at the picture. All right, so now we have a bacterial infection. We've got, let's see. The first thing that has to happen, we need to activate the helper T cells because we need the helper T cells to help activate the B cells. Just like we needed the helper T cells to help activate the cytotoxic T cells. So here we have an antigen-presenting cell that is displaying its foreign antigen from a bacteria on the surface using its MHC2 protein. Remember, MHC2 proteins can talk to only helper T cells. So there's a binding occurring between the helper T cell and the antigen on the antigen-presenting cell. This is going to activate the helper T cells, help the helper T cells make more of themselves, and start releasing a cytokine. And in this case, these helper T cells, these activated ones, will change slightly to a different type of helper T cell called a type 2 helper T cell. In cell-mediated response, it was a type 1 helper T cell. All right, so we have our activated type 2 helper T cell. Now its job is to activate the B cell. In this case, notice what the B cell is binding to. The B cell is binding to that bacterial antigen through the helper T cell, not through the antigen presenting cell. So we have our helper T cell, our B cells, which in this case are already displaying foreign antigen. This is what's really weird about B cells, is they can display the foreign antigen and not realize and not be activated yet. They need the help of the helper T cell. So the B cells are displaying the foreign antigen on their surface using their MHC2 protein, and that's how they can bind to these activated type 2 helper T cells. Once that binding occurs, activation of the B cell, part of that was the binding. The second part of it means co-stimulation by the interleukins that are produced by that newly activated helper T cell. So now we've activated the B cell. Here are the B cell receptors on its surface. They look like antibodies, and basically they are. When the B cell gets activated, it's going to do the same thing that cytotoxic T cell did. It's going to make more active effector B cells, and then it's going to make memory B cells for later. The effector or active B cells are known as plasma cells, and their jobs are basically to create and release the antibodies, which are basically the B cell receptors on that surface designed to respond and bind to that initial antigen. So when all is said and done here, we're creating a bunch of antibodies that will leave the lymph node and go do all those different things I described to you earlier. Not kill anything, but help other things kill those bacterial cells. But then we also have some memory cells for later, the next time you're exposed to that same bacteria. Okay. All right, so the last thing that this chapter kind of talks about is the primary response and the secondary response. Now, in the book, they're talking about these responses specifically with the antibody-mediated response. But please understand that these two responses occur even in the cell-mediated response. So what are these? So the primary response is the first time you're exposed. All this stuff is happening the first time you're exposed to a particular microbe. So if you're talking about the antibody-mediated response, let's use strep as our example. The first time you're exposed to that particular strep strain, all this stuff has to happen. The goal is to create antibodies that can bind to that strep bacteria and help get rid of it. Well, you can measure during this initial response, you can measure, well, how many antibodies are you making? Here. So you've made a couple different kinds of antibodies in response to that particular infection. Notice how much. It's enough to deal with the current infection. But remember, during this primary response, you're also creating memory cells. So the secondary response would be the second time you're exposed to that same microbe. You're still creating the same antibodies, but look how much more you're creating and at what time period. You're creating more antibodies a lot more quickly because you have all these memory cells now that you didn't have before. You can talk about primary and secondary response with respect to the cell-mediated response as well. But instead of looking at antibody levels, you're going to look at cytotoxic T cell levels. So during the primary response, these are cytotoxic T cell levels. The second time you're exposed, cytotoxic T cell levels are going to be higher faster because of the memory cells that you've made during the primary response. So don't equate primary and secondary response only with the antibody-mediated response. This is true for the cell-mediated response as well. All right. The last thing, and this comes up again in chapter 17. So chapter 17 is going to be talking about different types of immunizations. But basically, let's go back to that in a second. If you're looking at these two different responses, this is the basis behind vaccines. So when you get a vaccine, you're basically triggering a primary response, a primary adaptive immune response, whether it's a cell-mediated response or an antibody-mediated response. You're triggering that without actually getting sick. And what is the purpose of that? The purpose is to create the memory cells so that if you're actually exposed to a live infection, you already have memory cells and your secondary response will respond now and you won't get sick. So these types of immunity kind of have something to do with that. So naturally acquired immunity versus artificially acquired immunity. So naturally acquired immunity would be your body is responding to, there's a couple ways to think about that. So what's the best way to do this? Let's see. There's naturally acquired immunity and there's artificially acquired immunity, but there's also active versus passive. Let me start with active versus passive. Active immunity means your body is actually eliciting an immune response. Passive immunity means you are acquiring some of the key players from the immune system to like beef up an immune response, but you're not actually doing the immune response. So, and then naturally versus artificially means how do you get these things? So a naturally acquired active immune response is basically you getting an infection. So you've naturally acquired this foreign antigen, and now you are eliciting an immune response against it. And you create memory cells, so that's your immunity against it. So this is back when, before they had chickenpox vaccines, we used to have chickenpox parties where somebody in our class would get the chickenpox, and then everybody's parents would send their kids over to that kid's house so we could get it too, because it was better to have it as a kid than as an adult. All right. Naturally acquired passive immunity would be getting antibodies naturally. So like getting antibodies in breast milk, through breast milk, or even in utero across the placenta. So the baby is acquiring naturally created antibodies. So that's the natural part. But we call this passive immunity because the baby didn't actually go through an immune response and produce the antibodies herself. She got the antibodies from her mom. All right, now let's look at artificial. So artificially acquired active immunity would be a vaccine. It's active because your body is undergoing a primary immune response and producing memory cells in response to an antigen that's in the vaccine, but artificially acquired, meaning you're getting that antigen from your shot, not from the environment because somebody else was sick. Artificially acquired passive immunity would be antibiotics. So when you're given an antibiotic against a bacterial infection, you're basically given antibodies. You're beefing up your own antibody population. You're not producing those antibodies. You might be producing some on your own, but taking that amoxicillin just beefs up your numbers of those antibodies artificially. So it's passive because getting the antibodies does not trigger the immune response. You're just beefing up your own immune response, and it's artificial because you're getting it in the form of a pill. Okay? All right. I hope that helps a little bit. I realize that there is a lot of detail in these two different immune responses, but if you stick to the outline that I posted, those are the details that you have to know. So listen to this again if you need to. Use the pictures in the notes like I was using. Go to the other lectures that I posted by that other instructor. She might go through even more detail than you need to know, but when she draws things out as she talks, I think that might help, especially for you visual learners out there. So thanks for listening. It'll be posted to eCampus forever, so you can go back and look at that as many times as you need to. If you still have questions after you've looked at the study guide, make sure you post those to the class discussion board, and I'll try to answer them there. All right, thanks.